Good morning, dear students. Today, I am going to talk to you on taxonomy in relation to cytology or cytology in relation to taxonomy, or it is otherwise called as cytotaxonomy. This is for the botany students of Mysore University, 5th Sem, Unit 2. These are the contents which we will be discussing upon. Now, what is cytotaxonomy? Cytotaxonomy is a branch of taxonomy which uses the characteristics of cellular structures such as chromosomes to classify organisms. In other words, it is the application of cytological data in elucidation of taxonomic problems like distinguishing taxa, to determine the origin of groups, and to understand the evolutionary history of related taxa, particularly those at the infraspecific and specific levels. Infraspecific is below the species level and specific level is the species level. Improvement in the cytological techniques permit chromosomal studies. Various attributes like chromosome number, structure, behavior of chromosomes, and aberrations in reproduction is of great value in taxonomy, with chromosome number being the most widely used and quoted character. Chromosome numbers are usually determined at mitosis and quoted as the diploid number 2n unless dealing with a polyploid series. In that case, the base number or number of chromosomes in the genome of the original haploid is quoted. Another useful taxonomic character is the position of the centromere. So, cytological data is regarded as having more significance than other taxonomic evidence. This is a representative picture of cytotaxonomy. You can see the chromosomes under the microscope, anaphase stage. Now coming to the chromosome number. Chromosome number is usually constant for a species, and so it forms an important taxonomic character. According to some taxonomists, all taxa differing in chromosome number are to be regarded as distinct species. The gametophytic chromosome number of diploid species is designated as base number X. In diploids, N is equal to X. In polyploids, N is multiple of X. Example, in hexaploid species, 2N is equal to 6X and N is equal to 3X. In angiosperms, the chromosome number varies greatly. Example, we can see in Haplopappus gracilis, the chromosome number N is equal to 4. This is a plant belonging to Asteraceae. It is lowest chromosome number, and highest is 2N is equal to 265. 2N is equal to 4 in Haplopappus gracilis belonging to Asteraceae, and highest chromosome number in angiosperms is 2N is equal to 265 in a grass member, Poaceae member, Poa litosa. Closely related plants like the different species of a genus show chromosome numbers which reveal an arithmetic relation with one another, often in multiples of base numbers of characteristics of the genus. Example, different species of piper show chromosome numbers in multiples of 26, like 2n is equal to 52 in piper nigrum, 2n is equal to 78 in piper beetle, the beetle leaf, and 2n is equal to 104 in wild species of piper. This is the picture showing the chromosome evolution within Piperaceae. Darlington and Wiley in 1955, Love in 1977, etc. worked a lot on the chromosome number of various plants. International Association of Plant Taxonomy, IAPT, published an index to plant chromosome number in the series of Regnum Vegetabile, a book between 1967 to 1977 in 9 volumes. Diploid numbers are indicated as 2n, as you all know, and haploid as n. Now coming to the chromosome morphology. It is informative to the taxonomist in assessing affinities, the closeness and modes of origin of separate species. In genera like Crepis and Plantago, the large size and small number of their chromosomes have been of great value. These are some pictures showing the chromosome morphology of Crepis, large size, chromosome morphology of Plantago, etc. Chromosomes show variation in size, position of centromere and secondary constriction, etc. The structure of genome is a chromosome set in a species, is called karyotype, and its diagrammatic representation is called as an ideogram. Hutchinson separated Pandanus and Typha and Sporogonium on the basis of chromosome morphology and kept them under 2 different orders, Pandanales and Typhales. Next, coming to the chromosome size. Large chromosomes, low chromosome number and symmetrical karyotype represent a primitive status, while small chromosomes, high number and extreme asymmetry indicate advancement. So, if in a plant there is very less chromosomes, few chromosomes, and karyotype is symmetrical, that means the plant is primitive. So, if there are many number of small chromosomes and asymmetry in the karyotype, then it is an advanced species. The application of these principles has yielded very interesting results in Pandanales, Alismataceae, Hydrocaritaceae, Liliaceae, Amaryllidaceae and Dioscoriaceae. Even Cyperaceae and Juncaceae were separated due to distinct floral structure. They have holocentric chromosomes and hence now they are closely related or grouped together. The karyotype study of members of Agaveaceae confirms the shifting of Agave from Amaryllidaceae, inferior ovary, and Yucca from Liliaceae, superior ovary, into Agaveaceae. So now these both come under Agaveaceae. Earlier they were separated, they were in different families. So Agave was in Amaryllidaceae due to its inferior ovary and Yucca was in Liliaceae due to its superior ovary. But now the members of Agaveaceae, these both plants have 2 types of karyotypes consisting of 5 large and 25 small chromosomes. So based on this similarity, they have been placed together in Agaveaceae. So you can see the variation in size of chromosomes. See, they come of all sizes. Next, coming to the chromosome behavior in crosses. The behavior of chromosomes in crosses is a reliable factor in assessing relationships. Pathak in 1940 made a careful karyotypic analysis of various species of Agelopes, Cicale and Triticum, suggesting that the hexaploid Triticum spelta and Triticum vulgare were probably derived through hybridization between a tetraploid wheat. This is the chromosomes of the hexaploid Triticum spelta. Babcock in 1947 separated the closely related genera on the basis of chromosomal number and morphology. Youngia is separated from Crepis, while Tirotheca was merged with Crepis. Tragopogon myrus is tetraploid species as an amphidiploid, two diploid species from Tragopogon dubius and Tragopogon porifolius. The populations of infraspecific taxa showing different chromosomes number or morphology are taken as cytotypes. Rudall in 1997 suggested transfer of hosta belonging to Hostaceae, Camassia and Chlorogatum liliaceae to family Agaveaceae on the basis of bimodal karyotype. Judd in 2002 and Thorne in 2003 also supported the statement. So, based on cytology, we get an idea of the genetic relationships between plant populations. Cytology has made an outstanding contribution to taxonomy. The chromosome number, morphology, behavior at meiosis, polyploidy, chromosomal aberrations are all of considerable importance in solving many taxonomic problems. Cytological evidences are also used for distinguishing the taxa, to determine the origin of groups and to understand the evolutionary history of related taxa, particularly those at the infraspecific and specific levels. Infraspecific is below the species level, subspecies level. So, cytotaxonomy is a part of experimental taxonomy. Thank you.